Hi, everyone. I'm Solenne Correard, and today I'm going to talk to you about the variant catalog pipeline, which was temporarily known as CAFE. So if you've heard about CAFE, it's the same thing. But I'm actually also going to talk about inequity in healthcare. And first, I want to acknowledge that me and the other Westerman Lab members who worked on that project work, live, and play on the traditional, ancestral, and unceded territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations. So first, what is a variant catalog and why do we need it? I'm very glad we had the Illumina talk just before because I'm also all about DNA. So let's imagine you have this cohort of 5 individuals. They get their genome sequenced, and we identify variants, differences between those genomes. For example, at this position, some individual carry an A and some individual carry a C, and we can calculate the frequency. The A allele has a frequency of 006, and the C allele has a frequency of 004. And that's what a variant catalog is. It's just a list of variants and their frequency within a population. And why do we use it? So there's different use for a variant catalog, but I work for a Children's Hospital Research Institute, and so we mostly use it for diagnosis of rare disease. So if a kid comes to the hospital with a suspected rare disease, they often get offered whole exome or whole genome sequences. And so we get the data, and we start with a huge number of variants, and we have to filter them to try and identify the causal variant for their disease. So one of the first filters is to use variant catalogues to remove all the variants that are frequent in the population, because a variant frequent in the population is unlikely going to be responsible for a rare disease. Then we use other filters, such as family member, variant prediction, and candidate genes, and hopefully we narrow it down to 1 to 3 candidate variants. And we give the child and the family a diagnosis. But what happened in the project is that they found out that those number works for kids that have a European ancestry, for example. But when an indigenous kid comes to the hospital and they get whole genome and get the same process, unfortunately, because in the existing variant catalogs, there is no indigenous participants, the filtering is way less efficient. And so from the beginning, we have more we filter out less variants with the variant catalog, so even using family member, variant prediction, and candidate genes, we end up with more variants at the end. And 3 to 7 may seem like not a big difference, but it is. And it's harder to give a diagnosis to an indigenous kid. And that's clearly unfair, and that needed to be addressed. So the Silent Genomes Project was born. It's a very big project that aims at reducing healthcare disparities and improving diagnosis success for children with genetic diseases from indigenous population in Canada. And a small part of this project is to build the Indigenous Background Variant Library, also known as IBVL, which is a variant catalog for the indigenous population of Canada. And this is not a new idea. There is a lot of variant catalogs now around the world. The main one is Nomad. It's an aggregated initiative. They have a lot of participants with European ancestry, also African American and Latino ancestry. And some are more targeted towards some populations, such as the Iranian that work with the Iranian population. There is some project in Africa. There's a project in Brazil. So this concept is not new. And we're working very closely with Genomics Athera in New Zealand, who is doing a very similar project with the Maori population. And so we wanted to build this Indigenous Background Variant Library. And so the question was, how do we process the data? We're going to get about 1,000 genomes from indigenous participants. How do we get that into a list of variants and their frequency? We came up with 2 options at first. The one was to use Nomad pipeline. Nomad is the biggest variant catalog. They spend they did an amazing job at developing it. But their pipeline is configured toward cloud platforms, and as they said, not everybody can use cloud or won't use cloud. It's disparate because there's different classes of variants that may be responsible for rare disease. The obvious one are single nucleotide variants, but there is also mitochondrial variants or structural variants, short tandem repeats. And they added all of that over time, but not in an homogeneous way. And finally, it's in WDL, and we'll all agree that it's WDL. The other option was to use other pipeline, because as I said, other countries develop their own variant catalogs. But most of them rely on in-house script, not really pipeline that were published. Most of them just rely on GATK put together. And they are not comprehensive. They don't include all those variant classes. Most of them do only single nucleotide variants. So we had to create that variant catalog pipeline. And so we wanted it to rely on open access tool so that other groups could also use it later on. We wanted it to be comprehensive because every kid should get the best chances for diagnosis. So it was very important for us from the beginning that it would also include mitochondrial variants and structural variants. And it also had to be working on local servers. And we also wanted modularity, traceability, and reproducibility. So Nextflow was an obvious choice. And so this is the pipeline. I'm not asking you to read the name of the tools that are on the image, but the idea is that it takes FASTQ files at the input file. So as for now, it's designed toward Illumina paired and short read sequences. And it outputs a bunch of VCF that contains the list of variants, the frequency within the population, and some basic annotation. And the gray parts are independent from each other. So the first one is mapping, so it takes FASTQ and it outputs BAM files. The second part is for the mitochondrial variants, so it takes as input the BAM file, output it from the first part, and output VCF with mitochondrial variants. The third one is for single nucleotide variant, and the fourth is for structural variant, which also includes short tandem repeat and mobile element insertions. And the reason we wanted those parts to be independent is that some of the existing variant catalog only include single nucleotide variant. And we hope that by making that modular, they could just take part 2 and or part 4 and add mitochondrial variant and structural variants to their database. So the pipeline works. It was tested on 100 public genome on the local server at the institute I work in. And so I was very happy with that. And it's available. You can use the QR code, or you can go on the Wasserman Lab GitHub, and you'll find it under the name of Café Pipeline, which is going to change very soon. And we also put a paper out on bioRxiv if you want more details about the tools or the, really the characteristic of the pipeline. So as I said, it's wrote in Nextflow, and we want to NF-coreize it. So I started that during the hackathon, so thanks everyone for your help. But there is still a lot of work to do. There's need to create some modules and some sub-workflow. Some of them overlap with existing pipelines such as SAREC or rare disease. So not all of them needs to be created, but there is some. And we want to implement spreadsheet to deal with batches in case the sample comes as batches and we want to do several release. So if you're interested, please join the effort. There is a channel on Slack that is called Variant Catalog, or you can ask me. There's plenty of things to do, so everyone's welcome. And so the overall idea by putting it maybe in NF-core, or at least NF-coreizing it, is that all the population could use it to create new variant catalogues specific for their population and address inequities, because no kid should be left out, and also to improve current variant catalog by maybe adding classes of variants such as mitochondrial or structural variants, because once again, this is what's going to make diagnosis better. So the key takeaway from my talk is not that there is a new pipeline around the corner. It's actually how we can all work and how the work that we do matters toward addressing inequities and how we can do that. And I thought about 2 points over the last couple of days. And one of them is to work toward equity. And I've heard a lot about the FAIR principles, but I invite all of you to learn, or to, for those who don't know, about the CARE principle, and improving diversity in the data set that you're working on. And that's very complicated because it may require community engagement, which take years and resources, so it's not just a box that you will tick. It actually sometimes not possible, but at least it's important that all of us, we take the time to reflect on the work that we're doing and see how we could make things better. If you're at the beginning of a project, it's great, but even if you're halfway in or about to publish, it's really important that we all think about, are we inclusive? Can things be done differently next time? And the second thing is accessibility. So with NF-Core and Nextflow, it's obvious. Most of you are already doing it, but it's to use open source tools and make the pipelines available. It's good documentation. And again, thanks to the documentation team during the hackathon, because in most of our projects, we are lucky enough that we have bioinformaticians, we have people managing our samples, we have an IT support, but sometimes in other countries, they don't have all those resources, and it may just be one person who wants to do better for their community, and they must be able to do that. It may just be a clinician, so the documentation has to be really good so that they can also use the tool that we all work on. And offer several options to process the data, which include local processes and cloud processing. And I would like to finish by thanking all the organization for the great hackathon, the great summit, and giving me the opportunity to present this work, the CZI for giving me a travel bursary, and the Wasserman Lab, and especially with Wasserman, as well as Mohamed Abdallah and Brittany Ewitson, who helped with developing the variant catalog pipeline, and all the Silent project team members. Thank you.
Thank you for the talk, Solenne. We have a few questions here. So Saba is asking, what does the outreach look like for the children that are patients at this hospital?
Can you repeat? Sorry.
What does the outreach look like for the children that are patients at this hospital?
I'm not sure what do they mean by outreach. So
I think probably how these kids are they are taken care of. So, for example, for my master's degree, I worked with sarcoma in Brazil, and there was this huge issue that the kids had places to stay in the hospital, but the family that was taking care of them were not. And then there was some issues trying to help with that. So, how the kids and the families they are taken care of during these processes?
that's a very interesting question, and I really focused today's talk on the pipeline, but there is a huge work within the Silent Genome Project to increase accessibility. And as you mentioned, a lot of Indigenous families are in remote areas in Canada. Like I live in British Columbia, which is huge, and they may be in a remote area where there is no good internet connection, and sometimes they cannot get to the hospital. So a part of the project was actually to make things easier for them, so to try and do recruitment through like phone, and maybe trying to get them with their local health provider rather than asking them to come to the hospital. So there's been a lot of work toward also like increasing accessibility. I haven't worked on that personally. You can find a lot of information about that on the Silent Genome website, but it has been a big part of the project.
And Anne is asking, does the catalog also include HLA typing? HLA typing?
No. We actually had a team reaching out to us about that. No, but I'm happy to talk about it.
Thank you very much.
Thank you.